Hi, I'm Vincent Odekerken, and today we're talking about Parkinson's disease. This is the second video in a series of three discussing the different parts of what matters in Parkinson's disease. And now we're going to talk about pathophysiology. We discussed symptoms in the previous video, and I'll be discussing medical management in the next video. Why is this important? As Ray Dorsey and Bastian Bloom already very nicely put in a recent viewpoint, Parkinson's disease is a disease that continues to grow in numbers. Currently in the Netherlands, there are 30 or 35,000 people with Parkinson's disease, and this is a number that will likely double within the next 20 years if the current trend is maintained. So we need to prevent Parkinson's disease from happening if possible. We need to treat it well if it happens. As I discussed before, Parkinson's disease is not a single entity, but there are some pathological things that all patients have in common. It's misfolding of proteins, and specifically the protein alpha-synuclein plays a large role in this. Proteins have a 3D structure, and if that 3D structure is disaligned, it's harder for the body to get rid of those proteins because they don't fit in the regular garbage disposal area. So what happens? If proteins misfold, cells tend to dysfunction, and that dysfunction leads to cell death. If that cell death happens in the substantia nigra, which is depicted here in the mesocephalon as an actual cut of the brain, you get nigrostriatal degeneration. So the mesocephalonic substantia nigra degenerates, projections up to the basal ganglia degenerate, and you get motor symptoms in Parkinson's disease. If you look at in vivo or in pathological sections of this, you see healthy substantia nigra here as the black melanin-rich cells in the mesocephalon. In the PD patient, you see that the melanin is missing and there's a depigmentation, which correlates with degeneration of your substantia nigra. Also, if you look at it under the microscope, you see less melanin-rich cells, which means there's degeneration of the dopamine-producing cells in the substantia nigra. So, what happens on a molecular basis? Everybody has alpha-synuclein as part of a protein that you need for the function of your cells. Only it's folded in a way that it can be degraded and reused. But under certain circumstances, what we call native alpha-synuclein, just a random healthy bit of alpha-synuclein, can misfold, and those misfolded proteins are harder to get rid of. Those misfolded proteins can clump together into oligomers. Those are even harder to get rid of. You can get rid of these, and you have certain processes in the body that help get rid of these. Autophagy through phagosomes and lysosomes, and peptide breakdown through proteasomes. And this is all in a balance. So in a healthy balance, there's enough breakdown of these misfolded proteins and oligomers through phagosomal, lysosomal, and proteasomal action to get rid of the pathologically folded proteins. If there's too much production of misfolded proteins, or if there's a lack of breakdown, you get clutters of proteins within the cell. And those lead to dysfunction. If the clutter gets too big, the oligomers form amyloid fibrils, which is a larger clutter of oligomers sticking together, which is even harder to break down for the body because it's a more pathological state of those proteins. These fibrils lead to oxidative stress because the cell will dysfunction because the fibrils get in the way. Proteins get sequestrated, axonal transport gets disrupted, hence synapses start to dysfunction. Mitochondria within the cell core start to dysfunction, or within the cell body start to dysfunction. So the cell dysfunctions as a whole. And this leads to excessive cell death. Here you see a picture in which you see the different fibrils cluttering in the axon and in the dendrites near the cell core, causing to cellular dysfunction. And this is not limited to within the cell. Fibrils can be transmitted through dendrites or axons to other adjacent cells, leading to a spread of the pathological fibrils from cell to cell. If this goes on and on, more parts of the brain get affected by the pathologically folded proteins, and cellular dysfunction tends to get more frequent and more spread. So what happens, this is depigmentation of your nigrostriatal system with your substantia nigra, your synaptic projections from the substantia nigra to the basal ganglia, and dopamine that is released here to the basal ganglia network. If misfolded proteins lead to cell death within the substantia nigra, that usually happens when Parkinson's disease reaches the substantia nigra, because it doesn't start there, it just reaches it after a certain amount of years having the disease. The substantia nigra starts to degenerate, and if it starts to degenerate, the projections of the substantia nigra also degenerate. This leads to the motor symptoms of Parkinson's disease. But actually, the alpha-synuclein spreading is much wider than just the substantia nigra, only that is when we can make the diagnosis from a clinical viewpoint, because it leads to motor symptoms. It also explains that years before a clinical diagnosis, people can already have symptoms, dysautonomic syndromes, sleep symptoms, because other parts of the brain are affected before the substantia nigra is reached. We call it preclinical symptoms, but technically it's already clinical symptoms, but pre-motor symptoms. As the disease starts to spread further, other parts of the brain can get affected, and this can lead to more cortical dysfunction, such as cognitive disorders, postural instability, and balance disorders. But where does it originally come from, and how does it spread? The break stages of Parkinson's disease are something that are described for many and many years now, reaching eventually the cortex, but starting in the basal and mid-forebrain, hypothalamus and thalamus. But if you go even further back, you see that it enters the brain in your vagal nerve cores, and even before that, it's already present in the peripheral nervous system, mainly of your nose and your enteric nervous system, so your gut. And how does it get there? So we'd see that there's an influence of pathological or preformed fibrils, so pathological alpha-synuclein on regular alpha-synuclein. If there's a misfolding fibril nearby, it can affect healthy endogenous alpha-synuclein. And in some lab experiments, they have tested and proven that if you introduce those pathologically preformed fibrils to endogenous alpha-synuclein, it misfolds. So there is a bad influence of the exogenous PFF on your alpha-synuclein, leading to even more pathological clumps of alpha-synuclein. So if you look at a normal rat, and you inject them with the pathological exogenous PFF, you eventually see spread of this exogenous PFF and influence on endogenous alpha-synuclein, leading to Parkinson's disease. So you can introduce Parkinson's disease by spreading it in the gut. If you cut the vagal nerves and then introduce pathological exogenous PFF into the gut and into the enteric nervous system, it doesn't reach the brain, and they don't develop PD. Also, if you have genetically mutated the mice with a gene that affects the pathological creation of endogenous pathological alpha-synuclein, they also don't get alpha-synuclein formation and thus no pathological influence possible, so they don't develop PD. So this is interesting from a pathological viewpoint. So one of the newer theories that has been around for a while, but there's more and more evidence into this, is what's called the Vagal Highway, that the vagal nerve is one of the highways in which pathological alpha-synuclein reaches the brain. So what can be of influence of this? We know that your microbes in your enteric system can influence protein aggregation and formation of pathological clumps of alpha-synuclein. And if certain microbial agents are present, you have a higher chance of getting pathological alpha-synuclein in your gut and thus influence the vagal nerve alpha-synuclein and eventually reaching the brain. So your the different microbes in your gastrointestinal system are of influence on the chance of developing PD. And we also know that even as early as 20 years before the clinical diagnosis of PD, so clinical diagnosis being the motor symptoms, there can be pathological alpha-synuclein present in the enteric wall. If you perform a vagotomy, you can spread, it's harder to spread the pathological alpha-synuclein, so you slow the progression of the disease to the brain. And even in patients clinically, if you look at retrospective cohorts, people who have had a vagotomy for different reasons have a lower chance of developing PD than patients who have not had a vagotomy before any symptoms started. So that's if you had it in your early adulthood, there's a slightly less chance of developing PD compared to cohorts that didn't have vagotomies. This doesn't help if people already have PD because the disease has already reached the brain, so it's not a secondary prevention treatment that we can use. We know that certain types of infection influence the expression, and if you can get rid of that infection, the expression is lessened and your long-term chance of PD is probably lessened too. And interestingly, the appendix is a site in which the pathological isoforms of the preformed fibrils are more than elsewhere present, and that is also slightly correlated with the chance of PD. If people have had a appendectomy in their early childhood, their chance of developing PD are slightly less than patients who have never had a appendectomy. So that might be some sort of a very slight prevention of Parkinson's disease, which isn't useful from a total prevention viewpoint, but it's interesting as a theory. So take homes from this are alpha-synuclein, pathological alpha-synuclein can spread from cell to cell and from neuron to neuron. And aggregations of misfolded protein seem to be initiated in the gut and even after years and years only end up in the brain. So it's not just a brain disease, it's a gut to brain axis. If you look at the cell-to-cell promotion of misfolded proteins, that can also affect healthy proteins. That's kind of like what you see in prion diseases like variant Creutzfeldt-Jakob disease, only much slower and not transmittable from person to person. So what are risk factors that are known in PD or for the development of PD? If you look at external risk factors, there have been many studies which a lot can be said about the quality of the studies because you have to have very large groups. There's always some sort of publication bias or inclusion bias, but there are certain things that can be said about the increased risk of PD. So in red, we see some things that slightly increase your risk of getting PD. There is no absolute induction of PD. It's only slightly increasing your risk compared to a control population. The only absolute induction of Parkinson's disease is seen in PTP experiments. That's a drug that was introduced a couple of decades back was lab engineered and also very fast inductor of pathological alpha-synuclein forming and thus very rapid clinical Parkinson syndrome. That's only useful for lab settings right now. It's not clinically relevant. But what are known increased risk factors of PD? So use of pesticides, it's something that's been very well established long term. People who live in rural areas, especially farmers, back in the 70s, in which where the regulations weren't as strict on pesticide usage, have an increased risk of developing Parkinson's disease. It's also seen as a disease that is prevalent in farmers, especially in France. It's known as a farmer's disease, but it's worldwide spread, especially if pesticides are still used. So rural living, farming, and active use of pesticides increases your risk of Parkinson's disease, probably because it induces the forming of those pathological isoforms of alpha-synuclein, or it reduces the waste collection of it, so it reduces the breakdown of the pathological forms and thus leading to excessive cell death. Repeated head trauma, like boxers, is known to be a risk factor for PD. Muhammad Ali may be one of the hallmark patients in this category. Middle lifestyle, whatever that may be, is because it's a very broad category, it has an increased risk of getting PD slightly. High caloric diets slightly increase your risk of PD, and intake of lots of dairy can also slightly increase your risk of PD, but there's a lot of confounders that might be present in this set of risk factors. We also know that some things might reduce your risk of PD. Also, these numbers are a bit tricky, but we'll go through them. So the vagotomy is a strong relative risk reductor. Tonsillectomy, which also is a semi-vagal highway, just like the vagal nerve is, your tonsils also form a part of your ring of Waldeyer and thus also promote neuronal transport to the brain. Appendectomy we already discussed, but the figures are a bit two-sided in that. Smoking might reduce the risk of PD, but on the other hand, one might say that people who have less dopamine production tend to smoke less, so it's a chicken and egg discussion there too. Caffeine intake and NSAID use are also slightly correlated to a reduced risk of getting PD. So that's interesting. There's nothing that you can really do about it as of now, but there are some factors that help us lead in the etiological factors that we need to address to prevent PD eventually. So that's external risk factors. You also have internal risk factors, genetics. Most forms of Parkinson's disease don't run in families. Only 15% has a family history of first or second grade family having Parkinson's disease, and monogenetic Parkinson's disease, so one gene leading to the disease, is even more rare, which is only a couple of percent. The most common genes being LRRK2, PARK7, PINK1, PARKIN, and SNCA, which we discussed before. There are also risk genes that don't absolutely induce PD. The most interesting one being GBH1, which is also known in Gaucher's disease if you have it on both the alleles. So the GBH1 is particularly interesting because it might lead to a clue in prevention or treatment of progression of PD, because this is a gene that's used in a lot of places within the cell to maintain cell stability. It leads to, if you don't have it, to a gain of function near your endoplasmatic reticulum, leading to ER stress, which can induce alpha-synuclein aggregation. But it also leads to a loss of function, mainly in your lysosomal and Golgi function, decreasing autography and mitography, and thus decreasing your cell's ability to get rid of pathologically folded alpha-synuclein. So both in the gain of function and loss of function side, GBH1 seems to be a promoter of pathological alpha-synuclein within the cell. And there are some therapeutic interventions that you can use to reduce the gain of function or the loss of function. And this is still being investigated, but it's a very interesting way of looking at preventative therapies in GBH1 positive patients with Parkinson's disease. So, if you look at the pathophysiology as a whole, it's not a disease of your brain. It's probably a disease that starts in your gut and ends up in your brain years and years later. It has a prion-like distribution for your peripheral nervous system, in which the vagal nerve is the main culprit. And there are some risk factors, environmental, lifestyle, and genetic, that can influence the disease, but are only a very small proportion of the factors that promote Parkinson's disease, and it's not yet useful to use these lifestyle and environmental treatments as a preventative treatment. Apart from living healthy and without pesticides and living biologically might long-term reduce your risk of Parkinson's disease, but by how much we do not know yet. GBH1 is a specifically interesting genetic risk factor because it might be something that has a factor of disease prevention in a secondary way. So we're keeping a close tab on that in the future. This is all we have for today on the pathophysiology of Parkinson's disease. In my next video, I'll be covering medical management through medication and advanced therapies and lifestyle interventions. So keep close tabs on that. For now, thank you for your attention, and I'll see you next time.